Good morning, everyone. I am Mokles LCC. I go with the name CC as just told you. I'm working with Dr. Peter Herseth in the Homology Lab, Purdue University. The topic of my talk today is local effect of fruiting on different anatomical and physiological traits in apple. The general theme of my PhD thesis that I'm working on biennial bearing, studying biennial bearing. Why biennial bearing? We look at biennial bearing actually in a little bit different way. So it's not only why biennial bearing, but is biennial bearing really a way to know better about apple flowering in general? If you look at this picture here for golden delicious trees in the same orchard, the same row, find the first tree full of flowers and the rest of the trees with no flowers, clear bienniality. But you still can see within the first tree, which is full of flowers, some local spots with no flowers, and with the rest of the trees which are with no flowers, some spots full of flowers. So there is some kind of system, the whole system versus the local, the spur itself, effect on flower formation here. The current belief, which raised in the 1960s, that the seed-derived gibberellins move backward from the fruit back to the buds to inhibit flowering. And this actually was confirmed by measuring the GA's activity in the seeds, but was never confirmed by the gibberellin itself really moving backward to the buds. Another way to confirm this was by spraying exogenous gibberellins on the trees, but again, no one really confirmed that the gibberellins moves backward from the seeds to the buds. Leaf area is very important. Defoliation inhibit flowering. Partial and whole tree defoliation can inhibit flowering, and the effect of defoliation varies among cultivars. We need to focus a little bit on this point. The effect of defoliation varies among cultivars. If we think about what factors regulate flowering, it's vegetative growth, plant growth regulators, environmental stimuli, and developmental cause. If we think more where these things take place, most of them for sure will take place where? In the leaf. So leaves may affect the flowering in several ways. They provide carbohydrates, they provide plant growth regulators, they are receptors for the environmental signals, and they induce flowering genes. All of this will happen to encourage the spur to form flower. And sometimes this don't happen, or happen to inhibit flower formation. If we think about annual versus biennial, it's not only apple, it's most likely most of the fruit trees. You will find some cultivars with bienniality. So within every fruit tree, you can find differences among annual and biennial cultivars physiologically, and you can think that they respond in different way for flowering stimuli. So for us, it's one way to explore flowering pathway in apple is to identify the flowering promoting and or inhibiting signals in the leaves. The term spur leaves were always used to refer to all leaves on the spur. But if we look at this diagram, this is a spur. At the bottom, you will see spur leaves, which always formed early in the season. And after that, you will see the purse in the red circle, and this purse formed another leaves. So here we have the spur leaves, and we have the purse leaves. I just want to make sure that this is very clear, because we will use these terms a lot. Spur leaves, purse leaves, purse leaves, spur leaves. In my first year in my PhD, we started talking about a very simple way to see how these different leaves can impact flowering formation, spur leaves and purse leaves. So we designed an experiment in two cultivars, one annual and one biennial, to locally defoliate spur leaves, locally defoliate purse leaves, locally defoliate both spur leaves and purse leaves, and to leave both the spur leaves and purse leaves without defoliation. We did this 2 times, one time with fruiting on the with fruits on the spur, and one time with no fruits on the spur. So 2 cultivars and 8 treatments. And what we found out that most of the time the purse leaves, the local defoliation of the purse leaves, can inhibit flowering in Honeycrisp. We did this in 214, and we did this in 215 again, spring 215, the results. And you can see here that clearly it's off here, but you still can feel the same trend. So look at the control no fruit and the control fruit on the left side, and here's the control no fruit and control fruit. And we did this experiment, as I told you, in Honeycrisp and in Gala. So if you look at Gala, when we do this local defoliation, we still can form flowers. So you can locally defoliate a spur of Gala tree and it forms flower. And we repeated the same experiment again, and we still have the same trend. We decided to take this experiment one more step and to deeply study the physiological and anatomical traits of the leaves of the annual versus the perennial cultivars. Our objectives here is to clarify the differences and investigate the local effect of fruiting and study the effect of local fruiting on flowering, both in the annual and the perennial cultivars. What we hypothesize here, we think that these cultivars are different and they respond in different way to flowering stimuli. And also fruiting can have different local impact. This may impact flower formation. The methods, we picked 6 different cultivars, 3 annual and 3 biennial, Gala, Pink Lady, and Ruby Joan, with Honeycrisp, Fuji, and Golden Delicious. 4 trees per cultivar, 200 fruited spurs and 200 defruited spurs. And trees were manually thinned to medium crop load. All of these spurs were tagged with durable tags with numbers, so we can track them through the growing season. I know that this number 1 on this tree means what. We have 2 types of measurements here. The anatomical measurements, the spur leaf, the spur and the purse leaf number and area, the purse length, stomatic density, fruit weight, and fruit diameter. We have 6 sample dates through the growing season, and we have the end of the season. We have like 100 spur per everyone, per every cultivar, fruited and defruited. Another measurement is the leaf gas exchange parameter. We were lucky that Dr. Kerry Mitchell allowed us to use his portable photosynthesis device to measure this leaf gas exchange parameters. So we measured photosynthetic rate, transpiration rate, stomatal conductance, intercellular CO2, and water use efficiency. But how can we use that? We can't just go to the field and start measuring, right? We have to figure a way to measure these leaf gas exchange parameters. So the first step was to design a curve to see at what time of the day in megs, under our condition, these leaves are really active. Where is the peak? Where can we really measure? So we established this curve, and we figured out that from 9:30 a.m. to 2 p.m., and it was most of the field plants, you can measure these things. We pick the sunny day with 75 Fahrenheit temperature, and we choose the second fully expanded leaf per every spur. For this one, we had 3 fruited purses and 3 defruited purses per cultivar. So it's total 36 in almost 3 hours. We were riding the Kubota together and moving from tree to tree, from purse to purse, to take all of these measurements. Today, in the results, due to the time limitations, I can't present all the results. But what I will present, I will present the leaf gas exchange parameters. I will present the anatomical measurements, the stomatal density, the return bloom, and the seed number. I need to think more about seed number. So results. For the leaf gas exchange parameter, the proportion of total variation explained by bearing, fruiting, and their interaction. So on the left side, you will find these different parameters we measured. The first column, bearing and fruiting and their interaction. The first thing I want to mention here is not this strikes, but it's the fruiting. If you see here that fruiting don't really have part of affecting the variation between within these measurements. But what really have most of the influence is the bearing habit itself. And you see that it's highly significant in stomatal conductance and in transpiration. We will talk in details about stomatal conductance and in transpiration, but first I wanted to show you some trend we've noticed in the intercellular CO2 concentration, the CI, the first one. Even it's not really significant, but I think it was to share. If you look here at the left side, you will find Fuji, Honeycrisp, Golden Delicious. The red columns are fruited, and the gray are deep-fruited. So in the biennial cultivars, you always find that the concentration of intercellular CO2 is higher in the fruited versus the deep-fruited. So let's bring all the biennial together, and all the gala together, and all the annual together, and see how they look like. You see the fruited and deep-fruited are higher. Fruited is higher in biennial versus annual, in fruited versus deep-fruited within biennial, and is lower in fruited versus deep-fruited in annual. Here, if you bring all the biennial and all the annual together, you will find that in biennial, in general, the intercellular CO2 concentration is higher than the annual. For the stomatal conductance, fruited and deep-fruited in biennial, fruited and deep-fruited in annual, they are not different from each other, but they are different in the biennial with the annual. What does mean? The biennial and the annual, there's 98% variation is due to bienniality. And this goes also with transpiration. So transpiration, 99% of the variation is due to the bearing habit. Fruited, deep-fruited within biennial are the same. Fruited, deep-fruited within annual are the same. But fruited, deep-fruited in biennial and the annual, they are different from each other. The stomatal density. The first and the only anatomical traits we're going to talk about today, we found like 73% of the variation is due to the cultivar itself. But when you look deep in this, we found that Fuji is different only from Golden Delicious, Pink Lady, and Gala, but after that, all of them are the same. And if you look at the first three columns, the three biennial cultivars, and the last three columns, the three annual cultivars, they are almost the same. So there is no difference between the biennial and the annual cultivars in general in terms of the stomatal density. What we did in stomatal density, actually, we measured all types of leaves. We measured the spur fruited, the spur deep-fruited, the burst fruited, the burst deep-fruited, and shoot. Spur fruited and spur deep-fruited in all cultivars is almost the same. Burst fruited and burst deep-fruited in all cultivars is always the same. But most of the time you will find the shoot stomatal density is different than the burst and the spur. So it's very interesting here to notice that the burst deep-fruited versus shoot is significant in Pink Lady, Ruby Joan, Golden Delicious, and slightly significant in Fuji and Gala. Burst fruited versus shoot, significant in Gala, and slightly significant in Golden Delicious and Pink Lady. We look now at return bloom. Here is this trend, the fruited with F on the top of it is reduced comparing with the deep-fruited. This is in the biennial cultivar, it's very expected, but if you look at the annual cultivar, you will find most likely the same kind of relationship between fruited and deep-fruited. Seed number. Seed affect flowering formation, okay? Seed number in the annual and the biennial cultivars is almost the same with no significant effect, significant differences among them. Even Pink Lady shows a higher number, which is very annual cultivar. So in conclusion, fruiting appears to have local effect on flowering in both biennial and the annual cultivars. No difference in seed number between biennial and the annual cultivars. Different stomatal density among leaf types, but not associated with biennial bearing potential. So fruiting will have effect, but not because of the seed. Different stomatal density among cultivars, but not because of bienniality. The most important part here is that both stomatal conductance and transpiration are higher in biennial cultivars compared with annual cultivars, which is surprising. We think that higher xylem flow in biennial cultivars is puzzling, and we need to study xylem transported growth regulators. With this, I would like to acknowledge my committee members, Dr. Peter Hirst, my advisor, with Dr. Handa, Dr. Michael Bart, Dr. Bob Roth, the Apple team, those who are still with us and those who left us, Dr. Carrie Mitchell Lab and Dr. Carrie Mitchell himself, and the funding resource, the Egyptian government and Purdue University. And with this, I'll be so happy to receive any questions. Thank you.
I think we can squeeze a quick question in, but just one. Yes, Cheryl.
I've noticed that a number of triploid cultivars are quite alternate bearing, and they do not have functional seeds in general. So they would not be a source of gibberellin. Do you have any theory or comments on that?
Actually, how they how the idea of the effect of seeds on retained bloom was established in 1960s is by seedless cultivars. They made it seeded, and they compared seedless versus seeded, and they found that there's retained bloom in the seedless but not in the seeded. Okay, but if gibberellin really moves from the seed to the bud, this was never confirmed in any way.
But the Donegal, for example, is quite alternate bearing in its triploid, so the seeds are not really functional.
Yeah, I see what you mean.
So theoretically, it should be very annual bearing.
Yes, I see what you mean. That's if we think that the seeds are really the major source of inhibiting flowering, so this should come in the opposite way of what you are saying. But with what you are saying, you are confirming what we are saying, that it's not only the seeds. Maybe the seeds play a role, maybe an indirect role towards like the fruit weight, but it's not really this direct rule toward inhibit flowering.
Thanks, Cece.
Thank you.